Hey, can somebody confirm you can hear me?
We can hear you.
Thank you. Before we begin, are there any questions? My voice is off. Today is February 25th, and we are supposed to be finishing chapter 8 and starting chapter 14. I'm not sure we'll get there. We will get close to finishing chapter 8, but I'm not even sure we're going to finish chapter 8 tonight. Note that there is no lab schedule for today. I will be logging in for the first 15 minutes to answer questions. But other than that, there is no lab for today. Any questions about what we're doing? All right, let's go to chapter 8. We had finished talking about mutations. That we mentioned that a base mutation is some single base change. And didn't we talk about a base mutation last time? Or did I stop there?
No,
Oh,
you
remember,
stopped at missense.
I did talk about silent mutation, but I don't think I talked about, well, I did talk about a base mutation, but I haven't gone beyond that. Is that correct?
The
Yes,
page
this is
you're
the
on.
last.
All right. So a nonsense mutation is when you change one base, and instead of it changing from one codon to another codon, it changes to a stop codon. What's called a nonsense mutation. Let me blow this up. So previously we had the amino acids methionine, lysine, phenylalanine, glycine, and then the stop codon. Well, the AAA is changed to excuse me, it's not there. It's the TTC is changed to ATC, which changes from the amino acid lysine to the stop codon. And this really changes the protein because when you have a stop codon, you get a truncated protein. So the first part of the protein is correct, but then you get a truncated protein and the last part of the protein isn't even there. And this is called the nonsense mutation, where you have a truncated protein coming from the change in the amino acids in the DNA. Any questions about the base substitution, where we have a silent mutation, which simply changes the DNA, but it does not change the amino acid because the DNA codon still codes for the same amino acid. And if you remember, there are several amino acids that have more than one codon code for that same amino acid. So that's the silent mutation. And then there's the missense mutation, where you change the DNA and it changes the amino acid because it changes the codon. And then there's a nonsense mutation where you change the DNA and it codes for a stop codon. And instead of getting the protein, you get a truncated protein. Any questions about the base mutations? If not, let's talk about a frame shift mutation. A frame shift mutation is when you insert or delete a nucleotide base from the DNA. And it can be one or more bases that get altered, except if it is a multiple of three. And why we call this a frame shift mutation is when we remove one base, like here we had AAA, which coded for the amino acid phenylalanine, we then remove one base. and now we have AA, and that's not a codon, so then it reads the next nucleotide, and so it takes the C from the next codon, and that codes for a different amino acid. Instead of phenylalanine, we're now calling for leucine. But now the C is missing from this codon, and the codon's going to be CG, and then it'll borrow the next nucleotide from the next codon. And so all of the codons after the frameshift mutation are altered because the codon just shifts down one. And that can potentially change all of the amino acids in the protein. It may or may not because it depends on what the codon is, but it could potentially change all of the amino acids after the frameshift mutation. And that's why it's called the frameshift mutation, because when you make this mutation, not only may you change the amino acid at the site of the mutation, but you may change all the amino acids in the protein after that. So that's why it's called the frameshift mutation. Now, what happens if you remove 3 nucleotide bases? Like you take out all 3 of the A's, then the protein will be missing this one amino acid, phenylalanine, but the protein will be the same as before because the next nucleotide and the next, the next codon and the next codon will be correct. And that's why if you either add or remove 3 or a multiple of 3 bases, it's not a frameshift mutation. You do mutate the DNA, obviously, and you may mutate the protein. Usually you mutate the protein because we're pulling out an amino acid or inserting an amino acid. But the rest of the protein is correct. Any questions about the frameshift mutation? And I guess I should state that if you change the frameshift by 3 bases or a multiple of 3, it's not a frameshift mutation because it just makes a local change in that one or a few amino acids. And then the remainder of the protein will be the same. Any questions about frameshift mutation? Some Tay-Sachs patients are an example of a frameshift mutation, where either an extra DNA is found in the DNA or a DNA has been removed, a nucleotide has been. All right, so those are the different types of mutation. Let me remind you there's 3 types. There's a base substitution, and it can have 3 different types of base substitutions. There's a silent mutation, a missense mutation, and then a nonsense mutation. And then there's the frameshift mutation. And then there is the large area of DNA which is either added or removed, and that is called. Sorry, my brain isn't thinking. I took a nap and a macromutation. Any questions about the 3 types of mutations? All right. There can be different causes of mutations. There can be a spontaneous mutation, and it just happens at a certain rate just because the DNA is being replicated and the polymerase makes a mistake. So that mutation happens without anything causing it. And then there can be a mutagen, which can cause the mutation. There are two types of mutagens. There's radiation, and then there are chemical mutagens. Radiation has two types of radiation which can cause a mutation. There's ionizing radiation, and this is X-rays and gamma rays. This radiation has a lot of energy, and then when the energy hits an atom or a molecule, it gives the electron enough energy to knock that energy out of its orbit around the atom, meaning this electron then leaves the atom or the molecule. And that's why we call it ionizing radiation, because when that happens, this atom is now missing an electron, and that means this atom is an ion. It has a positive charge, because the electron has a negative charge. And this can be damaging to the genome because when it hits DNA, you damage the DNA by having the electron move. And if you hit another molecule like water, and then water bumps into the DNA, the water can steal an electron from the DNA. So in any case, ionizing radiation is bad for the cell. It creates ions that can react with the DNA, or you can hit the DNA directly, and then it damages the DNA. We're not going to talk about repair, but you can repair it. The other type of radiation that can damage DNA is non-ionizing radiation. Specifically, there's one type of non-ionizing radiation, and that is UV light. Because really, visible light is non-ionizing radiation, and visible light does not mutate the DNA. UV radiation does. UV radiation, when it hits DNA, it causes thymine dimers to occur when there are two thymines next to each other. So you have to have two thymines next to each other. UV light hits the two thymines, and then the thymines actually bind not once, but actually two times to each other. Thymine or any other DNA nucleotide does not have the nucleotide bases binding to each other. When UV light hits the thymine, they form a dimer where they actually bind to each other. This creates a bubble in the DNA, which this is trying to show you, because the thymines will not hydrogen bond with their corresponding adenines on the other strand of DNA. This means this region of the DNA cannot replicate, at least normally, and cannot transcribe the DNA, meaning the DNA polymerase will just stop right there. The DNA or the RNA polymerase will also stop right here if it's transcribing the DNA. The cell can repair that. We're not going to talk about it using light repair. And what it does is it notices the bubble there, and then it removes this region of the DNA around the bubble, and then it repairs this region. Any questions about how DNA can get damaged by UV light or by ionizing radiation? If not, let's briefly talk about chemical mutagens. Nitrous acid is a chemical mutagen, and the only chemical mutagen I'm going to talk about. When nitrous acid reacts with DNA, it can bind to this adenine right here, and then change the adenine slightly. So it's still an adenine, just an incorrect mutated adenine. And this mutated adenine doesn't act like a normal adenine. And when the DNA polymerase replicates this mutated adenine, it won't read it as an adenine, it'll read it as a guanine. And then it'll put a C next to the adenine. And then when this gets replicated, this will change the DNA from what used to be an AT sequence to a GC sequence. And that is how nitrous acid mutates DNA. It binds to the adenine and changes it so that the adenine, although it is still an adenine, just not a correct adenine, a mutated adenine, it won't read as an adenine, it'll read as a guanine. And then the DNA polymerase will put a C there. And that will eventually, when the DNA is replicated enough, let's see, is that 2 times, it'll change the DNA and you'll get this GC where it should have been an AT. Any questions about how nitrous acid mutates DNA and acts like a mutagen? All right. The frequency of spontaneous mutation is for every billion bases that are replicated, there will be one base which will be inserted by mistake. So it'll be the wrong base inserted. I think this is actually human DNA. I'm not sure if it's bacteria DNA or not. About 1,000 nucleotides make up one gene. So that means about 1 in 1,000,000 replicated genes will be mutated. What's the population of the Earth? Anyone know? Anyone guess? Is it more than 1,000,000?
More than 1 billion.
Yeah, it's around close to 8 billion, meaning many times more than 1,000,000. In fact, I think the city of Portland is close to 1,000,000, but I'm not sure about that. Don't quote me on that. It might be I'm thinking incorrectly, and maybe the population of the United States is around 6 million, something like that. Anyways, however you look at it, the population of the Earth is many times more than 1 million. So that means when a child is born someplace in the world, quite frequently, that child will be mutated. And it's not quite frequently because it's only one child in 1 million that's born will have a mutation in one gene of interest. Any question about any of that? So it's just telling you that we have a lot of nucleotides in our DNA. And if we look at one gene, the chance of that gene being mutated is not very great. But when we look at the human population, we see that there will be mutations born every day. And I don't know how many children are born each day, but it's probably close to around 1 million, but I don't know. It may be less than 1 million. I don't know. Like I said, the population of the Earth is around 8 billion. That I do know. Mutagens increase the mutation rate by 10 to 1,000 fold. And that increases the replicated genes being mutated from 1 in 1 million to 1 in 100,000 to as high as 1 in 1,000. And that's why mutagens are something you do not want to be exposed to. And you cigarette smokers should consider that, because cigarette smoke is the strongest mutagen that humans are normally exposed to. All right, how do we identify mutants experimentally? Like if someone were to see this walking down the street, everyone would say, yep, there goes a mutant, right? That is actually positive or direct selection, where we can detect a mutant because they either grow or appear differently. And normally we detect it on cells, so the cells either look differently or they grow differently. And that's positive or direct selection. You can also test for mutants by negative or indirect selection, where the mutant cells do not grow when they normally would. Let me explain that a little further. Negative selection being replica plating. I actually did this experiment when I was an undergraduate, where you get a plate that has isolated colonies. Then you get a velvet stamp and you stamp it into the plate. That puts the cells from the colonies on the velvet stamp. And then you velvet stamp the media plate here and a media plate there. This plate has media containing the amino acid histidine. This plate has media that lack the amino acid histidine. And then we look at the colonies that are growing, and this will be, let me see if I remember correctly. This one here will be this colony here, or that colony there. And then there's that colony there. And then there's this colony here, which is, I think that one there. And we did plate it there, but that colony did not grow. And it did grow in the amino acid that has, in the media that has the amino acid histidine. And that shows that this cell that formed a colony is a mutant colony. And for these cells to grow, you have to supply the amino acid histidine. Because if that amino acid is absent, the cells cannot grow. That's negative selection, able to detect a mutant from the fact that they're not growing in this media, but they will grow in media that has the amino acid histidine. Any questions about negative or positive selection for detecting a mutant? All right, let's change gears a little bit and talk about genetic recombination. Recombination is the exchange of genes between two DNA molecules. This is often from crossing over, where two homologous chromosomes, one from the mother, lowercase e or lowercase letters, and then one from the father, the uppercase letters. The two chromosomes actually come together in pair, and then they cross over. And then they break at the cross, and the father's capital E, capital F, capital G is there, and then we have the mother's lowercase h, lowercase I for the rest of the chromosome. And then the father's capital H, capital I is on the mother's chromosome. Although this is a recombined chromosome, so you can't really call it the mother's chromosome, but it's the remainder of the chromosome is the mother's chromosome. So that's crossing over. Crossing over is physically when the homologous chromosomes come together, pair up, cross over, and then break. And this happens all the time in mitosis and in meiosis, crossing over. Recombination is a little more general. I hope you understand that. It's an exchange of material between two DNA molecules. It can be crossing over, but it doesn't have to be. For example, let's say HI from this chromosome leaves this chromosome and then combines the chromosome A, which then has the lowercase h, I, and then the uppercase HI on it. So this would be now a bigger chromosome. That is recombination, but it is not crossing over. Everyone see that recombination is a little more general than crossing over? They are similar, and usually recombination is the same as crossing over, but recombination can be something different. Recombination is important because it creates new um, combinations of genes, puts new combinations of genes together on the chromosome. For example, before in the mother and father, we never had the capital G and the lowercase h together. And this could be something like the blue-eyed gene over here, and then the blonde hair gene over here. No, let's say dark hair over here. Yeah, that'd be better because it's capital. And then you can have in the offspring recombination happening by crossing over. And then the offspring's gametes can now put the brown-haired individual with the blue-eyed individuals, meaning the two genes are now linked together, making a reshuffling of the DNA in the population. Any question about any of that? Now, humans reproduce by sexual reproduction, and so that's a real shuffling of the genes in the population because not only do you have crossing over that can happen, forming the gametes, but you can also have, you also have half the offspring's DNA coming from the mother and half the offspring's DNA coming from the father. And so that's also a shuffling of the DNA in the population. And that's what all sexually reproducing organisms have. And that's probably an advantage because most organisms do reproduce sexually. Any questions about any of that? At least most higher organisms, I should say. Bacteria don't, but most higher organisms do. All right. So recombination only shuffles up the DNA that's currently present in the population. I should take a step back and state the only way you can get new genetic material in a population is by mutation, meaning if there is no gene for the allele or for the trait, then you have no chance of it occurring in the population. For example, dogs have yellow eyes and dogs have blue eyes and they have brown eyes. If there were to be a mutation in the human population creating yellow eyes, that would be by mutation, either spontaneously or chemically induced or radiation induced mutation. I'm not saying it's going to happen, but it could because dogs have the three eye colors. People could have the three eye colors too. And actually, if you go back in the human population more than 10,000 years ago, humans only had brownish eye color. There was no blue-eyed individuals in the human population more than 10,000 years ago. 10,000 years ago, in Northern Europe, there was a spontaneous mutation creating the blue-eyed allele. And then that mutation became the predominant allele in Northern Europe. And if you don't know, most of the Europeans at least have a significant portion of the population with the blue-eyed allele. But the blue-eyed allele is still very rare in Africa, at least southern Africa, south of the Sahara. It's rare in Australia, and it's also rare in Europe. It doesn't mean it can't happen. The Australian Aboriginals do have, in a very low number, the blue-eyed allele that was native to the Australian Aboriginal population, meaning it wasn't introduced by the Europeans when they settled Australia. Although I'm sure it was when there's reproduction, but my point is it did happen independently in Australia as well as it happened 10,000 years ago. And that's the first time it happened 10,000 years ago in Northern Europe. Any question about any of that? All right, where were we? Let's talk about genetic transfer. Genetic transfer is where a gene that was not existing in an individual or its offspring, uh, the gene can come in. And the gene can come in by three different ways. Obviously, four different ways if we're talking about spontaneous mutation. But there's genetic transfer that can happen. There's vertical gene transfer. This occurs during reproduction between generations of cells or between the parent and the offspring. And then there's horizontal gene transfer, where the genes move between cells of the same generation, meaning the DNA leaves the donor cell and then enters the recipient cell. Horizontal gene transfer can happen in one of three ways: by transformation, conjugation, and transduction. In transformation, DNA actually leaves a cell, and then you have naked DNA in the environment. Usually, how that happens is this other recipient cell that gives the DNA, it usually dies, at least in nature. And then we have the DNA out in the environment. The DNA can be incorporated by the recipient cell. And we're not going to talk about what's going on exactly, but that DNA can be brought into the cell. And then that lowercase a has to be recombined with the chromosome. And usually it recombines, and then the capital A, the original gene in the chromosome, is replaced by the lowercase a gene, which is brought into the cell. And then once that DNA from outside the cell, or a portion of it, is incorporated into the chromosome, we then have transformation occurring where this cell can now express a new gene which can change the phenotype of this cell. And that's the new gene is A that came into the cell and then came in and joined the chromosome, and now it's expressed, transforming the cell, because this cell will have a new property, the property encoded by the lowercase a gene. This is important for medical researchers or people doing what do you call it, work in the like the hospital environment, because what happens with the lowercase a gene codes for antibiotic resistance. This cell now gets the lowercase a gene. So this cell can change from being sensitive to that antibiotic to resistant to that antibiotic. And how important is that? Let me blow that up. All right. Spontaneous mutation. I'm just going to go spontaneous mutation. Happens at one in one gene, in one cell, will happen one time in 1 times 10 to the sixth. Let's go over. So in a million genes, you would have one spontaneous mutation. Transformation happens ten times more frequently, meaning you'll get transformation happening in 1 out of every 1 times 10 to the fifth bacteria cells. And so if you get antibiotic resistance occurring, which previously the cells had been sensitive to it, it could happen by spontaneous mutation, but it's only going to happen 1 out of a million times. Now, admittedly, usually when we have an infection, the minimum size would be a colony, which would be about 1 million cells. So you could have mutation by spontaneous mutation. But then again, it can happen by transformation, where the gene can come into the cells, which were previously sensitive, at 1 and 10 to the fifth cells, or 10 times more frequently than spontaneous mutation. What did I just do? There it is. Any questions about any of this? All right. Let's talk about, well, this is showing you transformation where the cell here takes up a piece of DNA, naked DNA in the environment, and then that changes the cell. The cell gets digestion from metabolizing that DNA, meaning transcribing that DNA, and then that can change the phenotype of the cell from what used to have smooth, I don't know, surface to a spiked surface or whatever. Let's talk about conjugation, sometimes called sexual conjugation. Sexual conjugation can happen in gram-negative cells when the F plus cell, which has a sex pilus, has the sex pilus skewer the F minus cell, which is a rounder cell lacking Fumbriae and the obviously lacking the sex pilus. And then the sex pilus literally skewers this cell, and then it acts as a winch to bring the two cells closer together. And then a plasmid from this cell can replicate and then move across the sex pilus into the F minus cell. Now let me state that the sex pilus is hollow, so the winch doesn't need to pull the cells right next together for sexual conjugation to happen, meaning for the DNA to move from this cell into that cell. If it were to stay like that, where this cell is quite a ways away from that cell, the plasmid could still move through the sex pilus from this cell to that cell. And we'll talk a little bit more about that in a little bit. In gram-positive cells, they don't have a sex pilus, but they have a mating bridge that can happen, and then sexual conjugation can happen because of the mating bridge. In the mating bridge,
We can't hear you.
Can't hear you.